Good morning, Mr. Krestensen. Good morning, ladies and gentlemen, family. I hereby open this academic ceremony in which Mr. Krestensen will defend the academic thesis entitled Innovations in Targeted and Untargeted Spatial Omics. Mr. Krestensen, may I invite you to present your major findings and conclusions in a brief presentation for the audience. Please, go ahead.
Thank you, Pro-Rector. Committee, supervisors, colleagues, friends, and family. Thank you all for coming today, where I'll be presenting some of my research that I've done in the past years for my PhD, titled Innovations in Targeted and Untargeted Spatial Omics. So I thought I would start with a bit of a definition slide on what is spatial omics, and if we start talking about omics to begin with. So omics is a sort of an all-surrounding word if we are looking at a given molecular species. So for example, DNA and the genome. We look at all the analysis points that we can do within this, and this encompasses then into genomics. The same can be done for proteomics, for proteins, or lipids for lipidomics. Now, if we take all of these things together, that is then what sort of encompasses biological complexity as we observe it in nature. And if we then look at the spatial part of spatial omics, that is then if we take, for example, a brain, an organ, and we make a coordinate system to look more in depth at what this brain looks like. And then we can pinpoint the molecular findings that we find from the omics into the coordinate system to see where these molecular findings happen. Now, the method that I use for this, for most of it, is called matrix-assisted laser desorption ionization mass spectrometry imaging, or MALDI-IHC for short. And this is a molecular imaging technique where you can image molecular species in a tissue. So if you have your tissue on a glass slide, as here, we apply then a matrix molecule to the tissue, which helps to extract a given molecule from the tissue. So this could be lipids, for example. The lipids are then fired at with a laser, and then the lipids are then shot into a mass spectrometer, which then detects everything that comes in there in an untargeted fashion. That gives us then a mass spectrum, as we can see here, where you have the intensity on the y-axis and the mass to charge, or the weight of all the molecules, on the x-axis. And what we can do then with this information is plot it in sort of a hotspot image where we have, in this case, we have two different tissues where each of the colors mapped out correspond with each of the, or one peak in the mass spectrum. Now for the first thing I want to talk about related to my thesis is chapter 3, where I investigated N-glycans for fresh frozen tissue. And N-glycans are these sugar molecules, so these things here are actually sugar molecules that are present in the cells and on proteins, and they're very important for a bunch of different mechanisms within the cell. So, for example, for protection from infections and recognition of self, as well as cell signaling. And these are actually highly influenced by both genetic and environmental factors, and they vary a lot between people, so there's a huge clinical implication in these molecules. The next part of this chapter relates to sample storage. So the samples that we analyze with MSI can either be formalin-fixed, paraffin-embedded, or FFPE samples, or they can be fresh frozen. So if we look at FFPE first, this is sort of the pathologist standard around the world, and that also means that they're highly available and they're easy to handle, which makes it, helps for the availability. Now, unfortunately, the chemicals used are very toxic, and actually they disrupt the native environment, the biology within the tissues, which is not good when we want to look at it in a molecular fashion. If we take a look at the fresh frozen tissue sample storage instead, this is very rapid preparation, because you just take your tissue and you dunk it in liquid nitrogen, and then they freeze immediately. And that actually keeps the biological environment intact, which is good for when we want to look at the molecular imagery in the tissue. However, there is quite a limited availability of this due to the storage facilities that you have to keep the samples very cold at -80 degrees. However, at M4I we have a good relationship with clinicians, and we therefore get a lot of fresh frozen samples into the lab. So that's why I wanted to investigate this. Now, if we want to look at glycans, it's also important that the glycans are released from their protein counterpart, so we can image them. And to do that, we use a cutting enzyme called pengase F. And to optimize this protocol or this method of releasing the glycans, we introduced 3 different steps into the method. So first off, there was an antigen retrieval where the tissue is heated up to let the glycans be more susceptible to the cutting. Then we use ice-cold reagents as well as a salty incubation chamber, so to keep the humidity and all the conditions very stable for the cutting. And immediately what we saw was a reduced molecular delocalization. So if you take a look at the 4 tissues on the left, this is FFPE tissue, and here you can see that actually the signal intensity of the glycans sort of spill out from the tissues themselves. Whereas if we look on the right at the fresh frozen tissue, you can see that the glycans or the molecules stay nicely within the boundaries of the tissue. What we also saw was more than 100-fold increase in signal compared to any sort of functioning fresh frozen protocol out there. So that is what you can see on the bottom here, where the top spectrum relates to the fresh frozen protocol, where all the single peaks relate to a single glycan. And then we actually also compared this new fresh frozen protocol to the old sort of gold standard, and we could see that the new protocol actually matches the FFPE gold standard and succeeds it in some ways, in that we were able to detect some glycans that were not detected in the FFPE protocol. And then we can say that we actually sort of extended the usability of MSI in clinical glycan research. Now, for the next part of the presentation, I will introduce a targeted imaging method called MALDI-IHC, or immunohistochemistry. And it's based on the principle of MSI. So you have here your standard MSI, where you do, for example, lipid imaging, and then you get all your untargeted images out of all the lipids that you detect in your sample. Now, if you want to make this into a targeted approach, you can use antibodies. And the antibodies we use go in and target specific proteins in your tissue, and they've been tagged with these mass tags that can be released after the antibodies have been bound. And then what we measure with mass spectrometry imaging again is the mass tags, and that allows for sort of a targeted multiplex imaging as a proxy for the proteins that we stained using the antibodies. Now, this was used for both chapter 4, 5, and 6. Where in chapter 4, I did a MALDI-IHC guided proteomic analysis in breast cancer. For chapter 5, I did a molecular profiling of glioblastoma cells. And for chapter 6, I looked at the interplay of immune cells and extracellular matrix in cancer metastasis. So going back to chapter 4 is when actually one of the first applications of MALDI-IHC in literature. And here we, of course, wanted to see if this multiplex imaging was functioning as it should. So we imaged a bunch of cancer markers in breast cancer, and these are markers for immune cells or invasion and metastasis and diagnostic markers. And they all mark different cells in the tissue, and they all have different functions. And that's also what we see then in the imagery, that the localization of the molecules differ between the different images. Now, what I wanted to do with this is take these images and look a bit more in depth. So look at the localization of the markers and look at the proteomic changes that happen to cause this altered imagery. And to do this, I took the MALDI-IHC images that I obtained and then did a computerized clustering, so clustering all the intensities into different clusters based on their localization, and then cut out the regions based on the clusters for sort of a follow-up untargeted proteomic analysis using mass spectrometry again. And what I got out with this, from this was 3 different clusters from the 6 different markers that we saw, where cluster 1 was related to tumor growth and survival, cluster 2 was related to immune activity and cancer metabolism, and cluster 3 was related to metastasis and inflammation. And with that, we can say that targeted MALDI-IHC was able to guide the untargeted proteomics and thus reveal spatially distinct tumor pathways in the cancer tissue. Now, moving to chapter 5 is when I wanted to look at the glioblastoma cells in sort of a co-culture fashion, where I combined targeted and untargeted imaging together. So first, I wanted to do lipid MSI of the cells, so look at the lipids that are present within the single cells. Then I wanted to do an imaging with MALDI-IHC to target the different cell types within the co-culture. So that could be glioblastoma cells, astrocytes, neurons, etc. And then I wanted to correlate the original lipid data with the cell type specific staining of MALDI-IHC to create a recognition model that was able to predict the cell types based on the lipid data alone. And what that looked like in practice is, as we can see here, so on the top we have the visualization of one lipid, and on the bottom a second lipid. And if we look within the green box or the green circle, we can see that lipid 2 is not present for this cell within the green circle, whereas lipid 1 is. And if we then go to the correlating MALDI-IHC staining, we can see that that is actually because it's a glioblastoma cell, where the other cells are neurons in this case. So we can say that lipid 2 is not present in the glioblastoma cells. This is one example out of many in the whole data set. So when we feed the whole mass spectrometry data into the recognition model, what we get out is a model that looks like this, that's able to distinguish different cell types within glioblastoma, astrocytes, and neurons based on their lipid data, and correctly predict them up to 93% of the cells present. Now, for the final chapter, I want to present some preliminary data because this is still an ongoing project, but it's a project looking at the correlation between the extracellular matrix, immune cells, and cancer markers. And we want to do multimodal imaging to look at this in breast cancer. And it's an ongoing collaboration actually with Johns Hopkins and the Medical University of South Carolina. And in the US, they want to look very specifically at the extracellular matrix and the collagens that are present there and how these change in the cancer and the metastasis. And what I did here was then do MALDI-IHC of different cancer markers and immune markers and also some extracellular matrix markers. And to do this, we had a patient cohort that we had access to where patients were followed for a long time, and we actually got a bunch of different samples from each patient, both relating to the primary tumor, metastatic sites, and control sites in the patients. And one of the things I want to highlight is how MALDI-IHC actually improves on pathology. So on the right, you can see a standard H&E staining where a pathologist has gone in and annotate different regions within the tumor core, where there is one region related to cancer, one to necrosis, and one to hepatocytes. However, if we take the corresponding MALDI-IHC staining, we can see that, for example, collagen 1A1 here, it actually crosses the different pathological barriers that were outlined by the pathologist. So we can say that it improves on pathology in this case. I could also see that MALDI-IHC was actually able to visualize a tumor treatment effect. So if we look at the top 5 tissues here, we can see that these are treated tumor tissues, and the bottom ones are untreated tumors. And here is a visualization of a tumor suppressor. So in this case, you can see that the tumor suppressor is present very much in the treated tumor, which is what we want to see from a biological point of view, of course. And if we look at a marker for metastasis, we can see that, on the other hand here, this one is more present in the untreated tumor than in the treated tumor itself. So in conclusion, we can say that Chapter 3 sort of invited this new method for N-glycans to help improve data availability and clinical relevance of glycans using MSI. For Chapter 4, we can say that novel spatially distinct tumor pathways, they furthered our fundamental understanding of breast cancer and can potentially lead to new treatment options. Chapter 5 showed how a targeted MALDI-IHC and untargeted MSI enables lipid-based cell type predictions and improves on single-cell differentiation. And Chapter 6 used targeted MSIs to help unravel molecular complexity across pathological barriers. And with that, I will hand the word back to the prorector.
Thank you very much for this clear and nicely illustrated presentation. Your supervisory team, Professor Heren and Drs. Belov and Anthony, have selected a team of experts that have, first of all, assessed the quality of your thesis. Their judgment was very positive. That's why you're now here to defend your thesis, because always, of course, questions remain, and we would like to see whether you can defend your thesis in an academic fashion. And that is why I now open the opposition by giving the word to Professor Platt. He's a professor in physiology of nutrition at the Maastricht University. Professor Platt, go ahead.
Thank you, Mr. Prorector. Dear candidate, I would like to start first by congratulating you with this great thesis. It's a nice piece of work, and of course, also congratulate your supervising team. It's a complex topic, at least for me it was complex, but you explained it as if it was easy. and the thesis, it was written in a very pleasant way. It was accessible and therefore it was easy to understand. I was especially fascinated by combining the techniques like the MSI and the immunohistochemistry, as you explained in your presentation. And I would like to go first to chapter 5, where you try to explain that the sample preparation and the MSI measurement has no effect in the ion intensities in the end. So I'm specifically looking to figure 3 at page 117. And there you compare the intensities that you present in panel C, D, and E, and you plotted their quantities in panel F. And I was wondering, I was a bit puzzled because in the legend you say this is the mean value of 10 single cells that were counted for each marker, while in the methods you talk about N is 4 subjects. So I was wondering, is this data coming from 4 subjects, or is this data from 1 subject where you used 10 cells to count the intensity? So can you explain a little bit more what you actually did?
Highly esteemed opponent, thank you for the kind words and for your question. So in this case, I think in the method, what I refer to is the number of slides in total that were analyzed to sort of go through this method and get it, eventually what we included as the results in the different images. That is from 4 different slides in total. And all of these slides, of course, have thousands of cells on there. So when I talk about then for figure 3, 10 different cells, is when I had to, of course, go and extract the molecular intensities from the cells. I had to go into each. So I chose one of the slides that was selected for these results. And then I selected the cells and extracted at randomly intensity from 10 of the cells out of the many cells present on the slide.
And those slices that you talk about, they were derived from 1 subject, 1 patient?
So I believe between, so not all of the sample slides that I used were from 1 subject. There were a few from 1. I think we had in total 5 different slides from each of the subjects that we got. And I think for this data, it was from a bunch of different subjects.
The reason that I'm asking is that I would like to challenge you a bit, because you like challenges. That's also in the title of your in one of the first pages. To challenge you a bit to design a kind of follow-up study, because we are always trying to push the boundaries and go a little bit further. So I'm a nutritionist, so I'm really interested in tracing the fluxes of bioactive compounds, so to say. So suppose that we could design a study together, and that we try to follow a certain bioactive compound, so a kind of sterile molecule. So it's not one of the lipids that you looked at, but let's say a sterile molecule. So how would such a study be designed? So we give some of these compounds to humans, and I want to trace the compounds in the intestinal wall. So just make a start, and now I will add in some additional questions. So how would you start such a study, and in terms of numbers of patients, and how reproducible is your method? So how can I be sure that if we look at 1 subject, it's the same in another subject?
That's a good question. So I would say that when we have to set up such a study, we have to.
So let's start with the panel of the immunohistochemistry markers in the intestinal wall. So we have enterocytes. We have goblet cells, Paneth cells, all these.
So of course we would select a panel that's relevant for the given question that we want to investigate, and then we want to make sure that, since you're talking about the patients or the investigated people, we want to make sure that these are controlled as much as possible. If we're looking at a given molecule, we want to make sure that as much as possible is kept the same between the subjects to not sort of induce unwanted differences that are not related to, say, the given drug that we want to look at, or the given marker that we want to look at. And if it's related to nutritional values, we want to keep them on a similar diet and make sure that there's no sort of underlying diseases that could maybe cause alterations that we're unaware of.
Yeah. So suppose that's all controlled for. So then we supply the sterols and we take the biopsies, so we have the material available. And then we are going to look at the cell structures, and we measure the quantity of the sterols. So how should we quantify those sterols? Because there's a lot of different sterols present, so we should try to identify the individual ones. Yeah. Do we need to label the sterols if we look at the dietary study?
So, assuming that you want to use the same method as I used, or then we would want to, if you want to be able to quantify it on a sort of on a way that we are able to quantify, we want to use some sort of internal standard to have a reference point, because if we just do an untargeted imaging like this, we cannot directly take the intensity and correlate it to the abundance that's actually in the cells, since it could be that in that case we can more look at the relevant differences, or the what is it called? The relative differences within a single person, but we cannot really compare it across different persons if we don't have a control to compare to. Yeah.
And talking about numbers of subjects, so how reproducible is the method in terms of variation? So if you do such a study, is it enough to include 1 or 2 subjects, or should we include larger
I would
numbers?
include a lot more, but I also know that it's difficult to recruit subjects for this. And
I would suppose that's not a problem. So what's the ideal situation?
The ideal situation would be, I would go more than 10, 20 people, as much as possible, because there will always be some variation, and we want to, of course, make this as small as possible. So the idea is when you then get more subjects, all the small variations they sort of fade away, and then you look at the large differences that are, then they come through more.
But can you say something more specific about the reproducibility? So if you do the measurement more than once, and so did you do some calculations about that? Can you, or is it just a guess that we say, well, let's take 12, or can we?
I didn't do any calculations for this, and it's been a while since I did any sort of power calculations. But I would have to look into that again. How to do that again?
I would be happy to look into that together, and I'm happy with the answers. And I give back the word to the prorector. Thank you very much.
Thank you, Professor Plat. The opposition will be continued by Professor Reis. She's a professor of analytics of biomolecular interactions at the Vrije Universiteit Amsterdam.
Thank you very much. So, dear candidate, I would like to congratulate you with a very nice thesis. It showed really that you wrote it with pleasure, and it also indeed made it that I read it with great pleasure. I also really enjoyed your everyday pictures of the lab rather than like the fancy equipment. It was actually very recognizable. I would like to discuss with you chapter 3 to start with on the N-glycosylation and the use of the on-tissue digestion. And I was actually wondering, how well can you retain your spatial information? So you mentioned, for example, on page 77 that, and also actually in your layman talk, that the glycans actually stay within the tissue boundaries, but you also want to retain your spatial resolution. So how do you prevent it from moving around?
Highly esteemed opponent, thank you for your nice comments and for the question. So one thing that we know about MSI is when we do the MALDI ionization, the molecules sort of stay in relative place to where they are desorbed from in the tissue. So that itself is sort of an assumption that we have, and when we then do the on-tissue digestion, what we want to avoid is, because you do the incubation in usually a very moist chamber, or sort of, it's a lot of homemade setups usually for the incubation chambers, unfortunately, but that can cause sort of condensation to fall down onto your tissue, and in this case, if water would fall down or any other solvent would fall down on your tissue, all the molecules would be washed away. And then when we keep the humidity in the incubation chamber very stable, we ensure that there is not too much condensation forming and eventually falling down onto your tissue.
So that's also why you keep everything at cold temperatures.
So everything is kept at cold temperatures, and everything is kept stable as possible within the incubation chamber. Yeah.
Would there be a way to test if things have been shifted or like moved around, or do you just rely on the resolution that you see and hope for the best?
Yeah, I guess it would be, what we also rely on a little bit is if the localization makes sense in sort of in a spatial, if we look at the tissue, if it makes sense to where it is when we do the imaging. So that is also something we take into consideration when we try to identify the glycans and say, okay, does it make sense that they're somewhat in this area of the tissue? Usually we have pathological and H&E stains of the tissues also. So that's something we can compare to. You could probably set up a control experiment where you could look at the delocalization, but it would maybe be difficult to measure the same tissue section both. Then you would have to create different environments of the incubation, and then do an imaging and see if there's a difference between, for example, consecutive sections of the tissue.
Oh, it is always a challenge. So I also want to discuss, you mentioned that you want to like multimodal techniques. So you do MS1 for spatial characterization, and then you move to LC-MS, so to digestion, full digestion, like so not to actually do the structural confirmation, etc. So you do them as two different experiments. Would it be possible to actually hyphenate everything in one? So do the MS1 followed by MS2 with, for example, other fragmentation techniques than digestion. So do like more top-down approach. What would be the pro or cons?
Of course, that would be possible if you have an instrument that would be able to do, a parallel analysis for MS2, for example. I would say that in the ideal world, it would be best to keep everything under one single experiment, because then you get rid of all the little changes that come between measurements. So one thing that can be a struggle in the lab is, of course, batch effect. So if you measure a lot of samples one day, they might not be the same the next day, but all the samples measured on the same day would be the same. Ideally, everything would be kept in one experiment.
So you mentioned also that you say ideally you would have an experiment or setup that does that, but I also read in your thesis you use the timsTOF flex. I was under the impression you could do those things, but maybe I'm wrong.
It does do that. And but when we were doing this, we had not quite sort of set up a method that was able to do this. So if we would have more time, it would have been ideal to do everything again and then do it under one single experiment. On this very instrument, yes, indeed.
I would like then to move to chapter 4, because I also really like the foldable, cleavable method and using this barcoding MALDI-IHC. Here also I have some more experimental questions regarding your experimental challenges. So I wanted to, for example, first discuss about the sensitivity. So what is the main challenge in the sensitivity, what you can see and not see with this method?
For MALDI-IHC?
Yeah.
So in the case presented here, we can see that the main difference in sensitivity probably comes from the peptides used. So the peptides themselves have sort of they all have a bit of a different structure, and I would assume that some peptides ionize better than other peptides, and that's also what causes the difference in the spectrum, as we see. So some of the peaks are very high compared to some of the other peaks, and when I've used different panels where they reuse the same peptide, so I can recognize the same mass across different peptides, usually there is a trend that the same peptides ionize better across different panels. And I think that is one of the things that would have to be really optimized if you want to do really high multiplexing of this. You have to make sure that all your peptides ionize.
Similar ionization
Exactly,
efficiency.
they should have a similar ionization efficiency, and also they should all be relatively easily ionizable. Yeah.
So to improve, if you would, for example, want to look at low abundant proteins, can you then make also, would it be possible to make antibodies that have more than one tag? Would it be beneficial, or would it actually be a problem for your method?
That would be a possibility, but then it would be, so in theory, if we just want to look at the localization of the molecules, this would not cause any issues. But one thing I also noticed is that it's hard to really compare the intensity also with the abundance of the protein. So that would really go out the window if we start using multiple peptides on a single antibody.
Mr. Pobrecht, is there still time for a small question? Or?
Small question.
Okay. Then we discussed the sensitivity. So on the spatial resolution, you have worked a lot on improving the spatial resolution by using ice-cold solutions and the matrix applications. So what is now the limiting factor at the moment, and what would you improve or suggest your supervisor team to improve if they would continue with this work? So it can be anything, also things that are very difficult.
I think what limits now is the instruments. They would have to follow along. So a lot of the commercial instruments are limited at 5 microns right now. But we have also in the lab sort of a transition mode source, for example, that can image down to lower spatial resolution. But I think that to commercialize that would be the limiting factor right now, because then otherwise you go into different sample preparation methods. Yeah.
Okay, thank you very much. Return the word.
Thank you, Professor Rijs. The opposition will be continued by Professor Korthals. He is a professor of analytical chemistry at the University of Amsterdam. Professor Korthals.
Thank you, Mr. Pro-Rector. Dear candidate, I love these images as well. And I thoroughly enjoyed reading your thesis. And I can only conclude that you really enjoyed it, and you're lucky to be amongst this group. And from reading what you did and the work that you did it, and from personal discussions, I can also see the real team player that you are. And that exists here, and that's very nice to see. I have a couple of questions related to Chapter 1, and then maybe one for Chapter 2, but there's a fair bit of focus on Chapter 4. But my first question to you is really on page 11. But before we go there, if you would talk about the technologies of proteomics, what comes to mind? Briefly.
Highly esteemed opponent. Thank you for the kind words and for the question. I want to quickly mention maybe that the photos are taken by the photographer here today as well. So he's the one behind all the pretty pictures. If we will go back to your questions, I think for proteomics, if I wanted to go into more proteomics research, I would probably focus on LCMS, liquid chromatography, mass spectrometry.
Because when you talk about, you go through a number of different technologies such as genomics, transcriptomics, proteomics, glycosylation, lipidomics, actually metabolomics. Then proteomics, you say on page 11, conventional gel electrophoresis, ELISAs, and protein microchips, but there's no mention of LCMS.
No,
Okay.
that's true.
But
I
you
wanted to make a bit of a story out of it. So I think at this point in the thesis, I didn't introduce mass spectrometry yet. And these methods that I mentioned are very traditional or, sort of used beforehand as a complementary to mass spectrometry.
On page 26, you have this figure 1, this one.
Yeah.
I tried to repeat your search and could never come up with the same numbers. I can only assume they're typos from me.
I hope so.
But if you look at the bar graph, what I see is it looks like there's some stagnation. And if we're looking at cost cutting, maybe we just stop developing with mass spectrometry imaging for the university? Because if I look at this here, there seems to be a trend that it's flattening off. Do you see that, or is it just me?
I think the part of the trend that you see is maybe related to the years around COVID, where less publications were just made in general because people could not go to the lab. So yeah, for 2021 and 2022, there's maybe less of a rise compared to the years before that.
Okay. That's fine as an answer, by the way.
Okay.
I'll take that. I also don't believe there's stagnation. And we need to move on to some, I don't know, more relevant questions. On page 89, I'll go to the BRCA study, the breast cancer. And I just need to check my notes here. Yeah, so this is FFPE data, and my question is here: have you looked at comparisons with FF data? Because this is a relatively small study, right? Two sets of tissues here. Has anything been? Do you know anything about if we look at FF and FFPE data on this rather important study? And then the extension of that, is there also LCMS data on these type of tissues?
So for this particular project, we only used FFPE data because this was, this was still early in sort of the method development of MALDI-IHC.
With your knowledge now about FF, and if you were in control of the money and had the same student like you, would you include that as well?
Yeah,
is it missing?
I would include it. I think after spending my years now in Ron's lab, I have a preference for fresh frozen tissue. And I believe that some of the modifications that we do with FFPE will inevitably change some molecular things in the tissue. I think I also mentioned in the end of the thesis that there is some degradation using both, like over time there's degradation of the tissue using both tissue types. But I would have a personal preference for fresh frozen tissue.
I just want to remark that's no criticism on any of the supervisors. They're all bigger than me, I think. Page 101, I like your wording. You say that there is interestingly, we're talking about the differences, and you say, interestingly, it's in the bottom paragraph under the images on about the second sentence. Interestingly, an inverse correlation was observed between measuring protein abundance with LCMS and targeted abundance with MALDI-IHC. So what's the go? What's happening here?
What's happening?
This is
disturbing. I think that this is something that would be interesting to look further into. What I personally think happens is that the antibody goes in and, of course, it binds to the proteins. And thereby, when the antibodies are bound, the trypsin that we use to digest then the proteins is not properly able to come in and cleave all the peptides that would otherwise be cleaved. So, and that would cause a direct inverse correlation, you could say, because all the proteins that you're actually looking for are kind of blocked by the antibodies.
Yeah, and this is just thinking of, what is optimal? Immunohistochemistry gives very high sensitivity. You have this new method. We have boosted sensitivity with MALDI-2. You didn't do a test or view the difference between just MALDI and MALDI-2, did you?
I believe there is a bit of a comparison between MALDI-1 and MALDI-2 on the previous page.
Oh, okay, righto. But then there's this difference. It's still kind of challenging to me. How would you be able to do quantitative analysis of these markers? What other method exists? I have one in mind, but maybe there are more.
If you want to be able to use the markers as a quantitative marker, you mean?
No, so you say interestingly, I'm going to call it disturbingly, there's a difference in quantitation in these two.
Yeah.
So how can we determine what is the real value here?
I think from just this, it's probably difficult to determine.
Yeah.
I would try to see if we could release the antibodies first and then have sort of a more clear image of the proteins in there.
Yeah. So I'll get to what I'm interested in is, could you with LCM take some of the tissue and do a targeted analysis, such as SRM analysis, and go to the clinical chemistry lab with that? Specifically looking for some of these peptide marker.
Yeah, that would also be an option to do it like that. But I think you would still, if the problem is on a tissue level, then if the problem is really because the antibodies are bound to the proteins, then that would still be an issue, I guess, in a SRM.
I also don't know. I'm just wondering if that was possible.
Yeah.
Okay, it's intriguing. Let's move on. So, I have 6 seconds left. Okay, just out of interest, I'll keep it short. Page 102, you talk about peaks detected. What I love about all of your work is that you really make this humanly interpretable in the sense that pathologists can actually understand the work that you're doing, and there's this wonderful translation. And then all of a sudden you talk about peaks. How many proteins were detected?
I don't remember the number off the top of my head.
I think we'll keep that for lunch.
Yeah.
Thank you very much.
Thank you, Professor Kortholt. Now the opposition will be continued by Dr. Leenaerts. She is Associate Professor in Gut Biology and Health at the Department of Surgery of this university. Please.
Dear candidate, so also my congratulations for this thesis, very nice work. It's also good for me to see what developments are there ongoing at your institute, so I can also learn from this and bring it to our samples from the patients, so that's very good. So I was having some questions about chapter 5. So there you have this cell culture approach.
Yeah.
Can you explain to me why did you choose for this approach and this for this study?
Highly esteemed opponent, thank you for your nice comments and for the question. The reason that this approach was chosen was that we had our collaborators for this project that were actually providing us with the cells beforehand, and then what Eva is somewhere here, has previously published is a way of growing the cells on the slide, and that was just what happened to be our tissue that we got.
Okay, and did you then also, so I missed a bit of the details on the cell culture. How long were these cells cultured? What was the approach?
I don't remember the exact details for that. So the cells were first cultured in a different lab, and then we got the cells from there, and then they were grown on ITO slides, and then we measured them. Yeah.
And did you also do some validation or verification that you also looked back into the original tissue, because there you have your spatial information, you have your tumor heterogeneity that you can assess. Did you compare these two and see whether your cell culture methods are really, that are no induced a lot of changes by just the culturing?
I did not look at the original data or the original tissues. I didn't have access to those prior to cell culturing. But that would be an interesting thing to look at. We did look at some, we did have access to RNA sequencing data to sort of compare a bit with the markers that we saw. That was sort of the only control that we had to the original data, so to say. That was not MSI.
Okay, and how how sensitive is the approach in terms of now you just culture the cells without any modification, without any differentiation towards a different cell type? Do you think that doing this, such a differentiation method, and see whether this translates really to these changes in your images or in your biological information that you capture with your method? Could that be done to really show, to really demonstrate that the technique is sensitive to pick up smaller differences between different culture techniques, different differentiation methods, etc.?
I actually don't know if this has been done, but I think it would be an interesting control to try to indeed force differentiation of different cell types in the culture and then see if we could pick up the differences between the unforced culture and the forced culture, because that is an assumption that is made now, indeed.
And the classification that you did, how confident were you, or what did you think of the results from that classification?
I would probably, ideally, had liked to repeated the experiments a few more times. Unfortunately, there was, no more tissue left, so that was a bit the limitation in this case. I think that what was done now was that the model was taught or was created on one slide and then tested on a separate slide. So it was cells grown on two different ITO slides. So that I'm pretty happy with already. But indeed, I would have liked to include more data into it to see if it could be more robust.
Okay, clear, thank you. And is this approach, would it be the best in terms of really characterizing cells and quantifying? So are there other methods that could be used besides this MSI that you did that could maybe give some more robust data, or do you have some ideas about that?
I think is normally used if you want to sort cells is FACS, for example. Which has been tested many times, and we know that works for sure. I think this is an interesting new way of using MSI, and ideally we want to have MSI be as broad and applicable as possible. And in this case, I tried to sort of broaden what is the possible applications of MSI. But I do think that, control experiments with FACS, or if we would be able to sort of control the number of different cells and which were exactly neurons and glioblastoma cells on a single slide would be interesting.
Thank you. So then I have some final question about chapter 6, because as you know, I have an interest in gut biology, and I was actually quite, I found your pictures on figure 6 of chapter 6 quite nice, where you have some markers stained in the small intestine and the colon. So I think it's very nice and also quite good resolution. So in a current project, I'm sampling also intestinal tissues from patients, and I'm interested in gut histology, gut and markers of gut barrier integrity and absorptive function. Do you think, so more like, to look for subtle differences between patient groups, do you think this approach would bring me something? Do you think it's good enough to also quantify maybe some smaller differences between groups? What do you think?
I think that indeed one of the issues right now is how we use this in a quantifiable way. And I know sort of the alternative would be a normal immunohistochemistry staining, or a staining that where we can actually, where we have a way to quantify the differences. So if you wanted to look at minute differences, we would have to be under the assumption that one antibody in one patient is comparable to the same antibody in a different patient.
But this seems, you think that this approach is, you say that's looking at one marker between tissues, that it is semi-quantified, or isn't it?
I think there is some things that point in that direction. I think, for example, in chapter 4, we look at this breast cancer patient with HER2, and there we can see that HER2, if it's HER2 positive, you have an upregulation of HER2. You don't have a complete, there's no HER2. And there we can see that there is a difference between the two, and it's not, so there is some sort of correlation with the intensity, but we don't exactly know what it is yet.
Okay, so this is some, there is maybe some area for improvement.
For sure.
But I think it's a nice technology. So I'm happy with your answers, and I give the word back to the Mr. Prorector.
Thank you, Dr. Leenaerts. And then finally, the opposition will be continued by Professor Biesen. He's a professor of experimental vascular pathology at this university, and he will finalize the opposition and defense. Please, go ahead.
I'm happy to do so. So, first of all, my congratulations on your excellent thesis. And I'd like to extend my wishes to the supervision team. It's not only a nicely formatted piece of work, which may reflect a very organized and rational mindset, virtues that are really important in science and that I unfortunately lack. But it also involves very important methodological work on Glycan detection, amongst others, and spatial transcriptomics. And I think this will push the field forward and also be benefit to the translation to diagnostic application. So I'm really happy to see that. And thereby, I'm also very happy to hear that your supervisor decided not to spend all the money that he received for a skiing tour in the Alps, but spend it on you, because it's mainly your virtue there. So, of course, I'm not an MS expert. I'm definitely not an expert on glioblastoma. I'm just a simple pathologist working with an affinity with macrophage biology, and even so on cardiovascular science. So that's beyond your topic, but I'll try to align with your background. And my questions, and that might not surprise you, relate to chapter 5, where you sort of developed and validated spatial omics and multiplex immunofluorescence integration. Multiplex MALDI. So the advantages of such integrated approach are obvious to me. You don't suffer from sort of alignment artifacts, and it's much faster probably, and it's also easier for pathologists to interpret in the end. Now, my first question relates to your antibody panel, and I've seen you tested, developed a whole set of antibodies and tagged them with these peptide probes, the photocleavable peptide probes, which I really like that approach. Now, looking into your table, that is table 1 on page 113, I see you included 5 markers for neurons. Now, if I look into figure 3, panel F, I see that of these 5 neuron markers, there are 2 that are sort of covering a pretty sizable area in your sections. SNCA and this amyloid peptide. The other three are not detectable. So can you explain this pretty massive difference between the 5 markers? Is that a technical artifact, or didn't they are they more difficult to ionize?
Highly esteemed opponent, thank you for the very kind words and for your questions. I think that this is a matter of, again, relating to maybe ionizability of the peptides used. So of course, as mentioned, though, the panel is developed elsewhere, and we bought it and used it, and assuming that everything was tested, because that's what they advertise. But then again, what we see again and again is that some of the peptides don't show up repeatedly, and actually around these masses, every time I've used them, they have not really ever shown up in a way that is comparable to some of the more really striking results. It could also be because you would have to use these in a higher concentration. So now I standardized the concentration, I think, to around 2 micrograms of antibody solution per milliliter. What could be done if you had unlimited amount of antibodies and money would be to try to test these and use just more antibody solution, especially for the ones that are detected in low intensity. Again, because we're not directly comparing intensities, then it wouldn't matter that we use a lot more antibodies and therefore peptides for these.
Can you think of a method to assess whether it's indeed a technical issue, or because there's also a pretty high degree of heterogeneity among neurons, as you know. So it might also be that the markers simply detect different phenotypes of these neurons. Can you think of a way to
so one way would be, for example, to sort out all the neurons and then do a proteomic analysis on these away from MSI, and in a way that we are sure to get a reliable view of the proteins present on the cells. But I think that would require, in this case, to wash away all the cells and collect them somewhere else.
Then another marker that you include is GLUT1, and that is a general glucose uptake channel. You indicate that it's specific for astrocytes. But unfortunately, it's not super specific for astrocytes. It can also stain endothelial cells, oligodendrocytes, and some other cell types. So that also is a bit of maybe a potential limitation of the way you analyze the data. You considered all the markers to be specific for a particular subset, and there might be a different way to analyze the data in a more integrated way, because actually the majority of these markers is not specific for a single subset. Could you think of a way to take advantage of the information of all the other markers for more precise phenotyping of the cell types that are contained in your section?
I think now that we have access to so many markers, an interesting thing would be to see where they overlap in cells and see which cells have multiple markers associated with a given cell type, and then use that to sort of corroborate our idea that this is a given cell. A whole other approach would be to develop a new antibody targeting a very specific protein on a cell.
You applied AI to analyze the data and to pinpoint with help of information that you obtained from the MALDI-MS to have a precise. would that also be, by extension, also be an approach that could be beneficial for a further improved assignment, annotation of the cell types? That you take advantage of all the information that's contained at MS layer, but also at IHC layer, and
yeah, for sure. So indeed, we could sort of extend the workflow to also include different AI models to then do morphological, look at how the cells are morphologically different and see how they then differ, and then use that also as a cell typing technique.
okay, thank you.
Thank you, Professor Biesen. Mr. Krestensen, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and the quality of your defense, and I request that you and your company await the results of our deliberations and our return in this room. Mr. Krestensen, the degree committee here present has assessed the quality of your thesis and the quality of your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Heren is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Kasper Kaagaard Krestensen, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other committee members and affixed with the official seal of the university. Dear Dr. Krestensen, dear Kasper, congratulations. After years of outstanding research and training, you've now crossed the finish line and have been honored as a new doctor. A doctorate that is incredibly well deserved as the rewards for all the efforts, energy and endurance you've put in along the way. You started in November 2020 when the world was recovering or experiencing a pandemic, and the future of the type of research you wanted to do was not at all clear. At that time, I had just received the NWO Physics Valorization Prize and could appoint a PhD on any research topic I would like, or take the group skiing. Yeah, we've heard that already. Sorry, group. We were already connected at that time, as I was supervising you when you were doing your master thesis research in the lab of Judith Steen in Harvard, working on models on protein aggregation in neurodegenerative disorders. It was part of your master at the Faculty of Psychology and Neuroscience of this university, and that's where you actually had an interest in neurobiology. Already then you stood out in that process by your diligence experimentally and your drive to get things done, and we started talking about this PhD position. In the end, you wanted to further develop your omics and imaging skills, and as we've heard today, you've done an outstanding job in reaching that goal. You truly became an expert in both targeted and untargeted imaging. One thing was clear at that time: you were incredibly smart, and you've shown that today as well. And you knew what you wanted, and I love working with people like that. So it was an easy hire. Now, part of a PhD is education. And you've also augmented your education in, among others, the Dubrovnik Summer School. There you did not only impress with the science you presented, but also gave me a reality check when I took a group hiking up the mountain, and you and one of your opponents, Gary, came running past us. So we were huffing and puffing up, and you went up there. It turned out you actually are good at running uphill in this, what you call the Frankenstein spaceship of MRI, and a quality that has really helped you along the way. So as scientists and recreational athletes, we have several common interests. And I could not resist to use some of these similarities in this laudatio. I did a side-by-side comparison on Strava, and you beat me on all athletic performance specs. Clearly, you have the advantage of youth. You also are a more multidisciplinary athlete with a triathletic swim, run, bike combination, where I solely bike a lot. Interestingly, you also seem more efficient, as in our all-time stats comparison. You have fewer activities and shorter distance, but more elevation gain, and accomplish all of your efforts in half the time that I do. So probably my age again. But in my mind, this also reflects your approach to science. During your PhD, you've tackled several different imaging and omics approaches that made you this truly skilled, multidisciplinary scientist. And your presentation today was a beautiful evidence piece of that. You've always focused on experimental efficiency by ensuring optimal experimental design, by absorbing knowledge presented to you by others. You also always, like in your athletic endeavors, kept your eyes on the prize, better papers and thesis chapters that we discussed today. So in working with you, I've also learned that the Danes and the Dutch are also very similar. They're both easygoing, often informal when they talk to each other. They say what they think, and open discussions are an important part of both cultures. A differentiator is that Danes apparently have a more drier sense of humor than the Dutch, which can be quite, sarcastic at sometimes, and they often make fun of authorities. I've probably been at the receiving end of that a couple of times. Yeah. I see you nodding maybe a little bit already. You were always an incredibly stable personality, or at least gave that impression to many of your colleagues. You remained calm and composed, like in this defense today, even when things were not going according to plan. As a result, a great personality to work with. Running up the hill, when collaborators were not providing the needed information and questions, was a challenge at times, but you impressively kept on moving, kept your cool, and moved on. Together with your paranymph Andre, you worked on optimizing the Glycan digestion protocol, and that has resulted in a much more stable approach in all of our proteolytic digestion studies, because we now knew what the important factors are that limit this, diffusion and sample prep. The two of you figured out what the real problems were, and as a result, a next generation of students can now perform more robust experiments. That is exactly what team science is about, and you excel at it. Core to these achievements is also the further development of the targeted MSI using MALDI-IHC in close collaboration with the Ambergen team. Something you took to some extent to MFRI after a discussion in Mannheim with the Ambergen guys, and reminded me back home of that this was something worth pursuing. From the unique combination of targeted and untargeted omics to single-cell MALDI-IHC, to its translational applications on single-patient TMAs, together with Christine Glunde and Peggy Angel in the States, you started the movement. Your methods are now employed in many studies that would not have been possible without the work described in your thesis. And also make up some of the amazing pictures you've put in your book and showing the diversity and mess a lab sometimes is when you move ahead science. Throughout your journey, you've been seconded by your two co-promoters that have provided your feedback on the progress manuscript and provided some of the daily supervision when I was traveling or lecturing outside the UM. I want to thank both Benjamin and Ian for their contributions to the work. The Valorization Prize idea of doing imaging proteomics on the mass microscope together with Ian has not yet materialized, but we are very close as we speak. So it will come. Pass your time. Your thesis is a perfect example of how things come together in ways that we could not have planned in advance. It was your drive and determination, fed by the environment that you helped to create, ultimately resulted in your success of today. It does make me wonder, what is harder, completing a triathlon or a PhD degree? I would never try to complete a triathlon, but you can give me your opinion over a drink during the celebrations of today. Kasper, I truly enjoyed supervising such an independent researcher and pleasant personality that now stands at the beginning of a new and unknown phase in his career. You do not know yet what the next step in your career will be, but the truth is, you've already built the most valuable thing of all, the ability to learn, adapt, create meaning, and have fun where you go. So, Dr. Krestensen, I'm honored to be the first one to congratulate you on this newly obtained honor. And of course, I want to extend my congratulations to your family, who's present here today, who probably were your greatest supporters throughout this journey. Now, it's time to celebrate. Congratulations.
Highly esteemed Dr. Krestensen, it's my great honor to congratulate you on behalf of the university, the Maastricht University, and the Board of Deans with the degree that you have just acquired. I also would like, of course, to congratulate your supervisory team, Professor Heren, Drs. Bellof and Anthony. Family, of course, your parents, family. And also I would like to congratulate your collaborators with the work that has been published in your thesis and in some publications. And it's obvious from the overview of your collaborators that you are a real team player, and it has already been mentioned by your promoter. I'm allowed to disclose a few remarks and comments that were made during our deliberations. It's obvious that your work is very important. Your results are important in pushing forward the field also in the direction of diagnostic application. Most of the chapters that you have that are in the thesis have been published, and I'm sure that also Chapter 6 will find its way to a high-ranking international scientific journal. The thesis covers a broad area of techniques and a broad area of biological questions. So it's a very multidisciplinary approach. With respect to the defense, I must say you defended your work in a real academic fashion. You were honest, very thoughtful, and I had the idea that also after a little while you became quite relaxed. And also what is important for a scientific discussion is that you expressed the limitations. You mentioned the limitations of your studies. And I think that all proves that you have developed into an independent academic researcher. Making a comparison to the sports that you perform, I had a recent PhD student who ran marathons. And I think you have experienced what she experienced also. And because she said the PhD is like a marathon, but in a PhD, the finish line seems to constantly move forward. Anyhow, congratulations again, and I wish you a lot of luck and happiness in your future life, of course, and lots of success in finding a new position. I understand you would like to work in Danish industry, so I wish you luck in finding a new career. I have a few announcements to make before I end this ceremony. I would like to ask the audience, except for the family, to go to the rafter for the reception. I would like to ask the family to join us, because we, as the team, would like to make some pictures in the staircase, the stairway in the front of the building, and I would like to also you to join us in that. Having said so, I close this academic ceremony.